Good morning. It is my pleasure to introduce to you John Maris, a true giant in the field of pediatric oncology for our Frontiers in Oncology speaker today. John is a Giulio D'Angio Professor of Pediatrics in the Perelman School of Medicine at Penn and at the Children's Hospital Philadelphia. John has focused his career over 3 decades on neuroblastoma, the most common extracranial solid tumor of childhood, with the dual goals of improving patient outcomes and using this disease as a model to understand cancer in general. As a card-carrying genomicist, his group has discovered several neuroblastoma susceptibility genes and has identified many of the oncogenic drivers in neuroblastoma. Dr. Maris has steadfastly sought to translate his discoveries to the clinic using precision medicine approaches, and as such, he is an internationally recognized practicing pediatric oncologist who cares for children with refractory neuroblastoma from around the world. In addition to being a spectacular and prolific scientist and a dedicated clinician, John is a gifted mentor and a born leader. He led the Neuroblastoma Target Project, the neuroblastoma portion of the pediatric preclinical testing program. And since 2013, he has co-led with me the St. Baldricks Stand Up to Cancer Pediatric Cancer Dream Team. Through this work, he has stretched his comfort zone to embrace immunology, and his group is now contributing remarkable insights into the role of self-antigens as potential immune targets for neuroblastoma. These are likely to be paradigm shifting for cancer in general. John also co-leads the Bo Biden Moonshot Cancer Center Award in Pediatric Immunotherapy. He holds a National Cancer Institute Outstanding Investigator Award, and he has received several prestigious awards, including election to the American Society of Clinical Investigation, the Oskey Award for Outstanding Pediatric Oncologists, and the Berwick Award at Penn for melding basic and clinical teaching, and the William Osler Patient-Oriented Research Award at Penn. I could go on about John's accomplishments. They are many. But mostly I want to say that he is an incredibly valued colleague and friend whom I have had the true privilege to work with and learn from over the last decades. We're excited to hear today from him through his presentation entitled Leveraging Developmental Biology to Develop Specific Immunotherapies for Childhood Cancers. Thank you.
Well, thank you very much, Crystal. That was really a very humbling introduction, and I very much appreciate it. So let me just grab a laser pointer. I'm very sorry I cannot be there in person, have my nice Stanford background, and very much appreciate being able to speak to you today in this virtual forum, and hopefully we'll have some time for discussion. I just wanted to disclose that I am a founder and have some equity interest in a company called Mutation Bio, and some of the technology that's in that company I'll speak about today. And I advise for a couple of companies, but none of that has anything to do with what I'm going to talk about today. So childhood cancers in the latter half of the in the 1980s, 1990s, really led the way in cancer in terms of improving outcomes, and largely through the empiric mixing and matching of chemotherapies and cooperation in clinical trials. But Crystal and I and many others have realized over the last 20, 25 years that we've really been kind of stuck. And this group of Kaplan-Meier curves look like your standard adult epithelial malignancy Kaplan-Meier curves, because these are all from children starting at the time of relapse. And so not only is relapse disease in childhood cancers lethal, we're not improving our cure rates much, if any, and for some diseases we haven't improved it at all. And survivors have significant lifelong comorbidities from the cytotoxic therapy. So clearly we need to do better. Crystal and I talked a decade ago about how childhood cancers are not little adult cancers in terms of immune evasion and how we're going to think about developing immunotherapy. There's obviously very little, if any, environmental influence, so therefore these tumors classically have very low mutation burdens. Crystal and others have shown pretty definitively that immune checkpoint inhibitors have very limited activity, at least in being used in an agnostic fashion. And of course, the childhood immune system is naive compared to an adult immune system that might be getting a little exhausted. And what's on the cell surface of childhood cancers is very different than the epithelial malignancies. And as I'll harp on in this talk several times, the developmental origins of the cancer are what really drive what could be good targets for our immunotherapies. And I think the only thing that is common between adult and pediatric malignancies is that we don't really have enough good immunotherapeutic targets. And that's something that we really would like to address. So Crystal mentioned that back in 2013, we were given the remarkable privilege of putting together a North American-based team. And really the main assumption of this team was that we really need to think from square one of what is the immunotherapeutic opportunities in childhood cancers, and it wouldn't be a trickle down from the adult malignancies. And now here in 2021, this initial effort funded through Stand Up to Cancer and St. Baldricks has grown into a continuation of St. Baldricks funding matched with our institutions, as well as a clinical trials network that Crystal runs, and this pediatric immunotherapy discovery and development network. And really this is where we like to do our fundamental discoveries, translate them to clinical trials throughout this collaborative consortium. And very proud of what this team has done over the years. And I can't even begin to speak to all that it's achieved, but I'm just going to give you a couple of anecdotes from my own lab, but would like to people in the audience to remember there's a lot that's come out of Crystal's lab and others at Stanford, and many of the other institutions around that I think is really making a big difference in pediatric cancer. So neuroblastoma is a childhood cancer of the sympathetic nervous system. So we've known for years that this disease arises from a misappropriation of normal autonomic neurodevelopmental pathways. It's a true childhood cancer in that these are toddlers who tend to get the disease. It represents a disproportionate amount of childhood cancer mortality. And the thing that drew me to the field, and I'll show on the next slide, is the wildly different clinical phenotypes that we see and really challenge us both from the laboratory and clinical perspective. What has, there's been tremendous progress in understanding the development of this sympathetic nervous system. It arises from the neural crest of the developing embryo. And we now have a very advanced understanding of the transcriptional programs that drive these to these final lineage fates. And it's now clear that the neuroblastomas arise somewhere during this bifurcation between a committed autonomic and mesenchymal. And it's now known quite clearly that these different cell sates can transition in a child's tumor. And this is an important aspect of developing immunotherapies that I won't have time to get into today, but could discuss in the question and answer if there's time. So this is what I mean by the clinical diversity. This is a child with this so-called famous 4S neuroblastoma described by Audrey Evans and Dan DiAngio in Philadelphia in the 1970s, where all of this blue stuff is tumor in the child's subcutaneous tissues. This child had a liver full of tumor, an adrenal gland that had tumor in it, and all of this just went away spontaneously with no chemo or radiotherapy. And we're still trying to figure out the mechanism that makes this complete spontaneous regression. And it is potentially very likely that the immune system has a lot to do with it. The problem is that the majority of neuroblastoma cases have these sort of very large tumors. Neuroblastoma of all human cancer is the one that is most widely metastatic at diagnosis, this special MIBG scan that shows these black spots everywhere. This is not just a bad case I selected for this. This is typical that the disease is incredibly widespread. And so this highly aggressive Burkitt's-like phenotype is something that is quite a challenge and is the reason why we need to develop new therapies. As Crystal said, I'm a trained geneticist, so over the years we've understood a lot of what makes a neuroblastoma tick. About 40% of high-risk cases have this so-called MYC amplification, literally hundreds of extra copies of the gene compared to the 15 or 20 copies that you get with HER2 amplification in breast. Yael Mosé described gain-of-function mutations in the ALK kinase, and inhibitors of this kinase have moved through phase 1 to now current phase 3 to hopefully layer in kinase inhibition for the patients that have these mutations at diagnosis. But what all cases have are these overexpression of key transcription factors that are critical to that, what I showed on this prior slide, those cell fate decisions. So these transcription factors get epigenetically deregulated in all neuroblastomas, and they give the proliferation signal and the anti-differentiation signals that are essential for sustaining the cancer phenotype. So in one hand, while it would be great to have a MYC drug, another would be great to have a drug against one of these, because what's shown with CRISPR is if you CRISPR out any one of these in a cancer cell, this whole network collapses and the cells die. So, but going after transcription factors has been a challenge, but something that we're trying to address in this program, and I'll end with that story. Sam Bahati had a really nice paper recently where he looked at a bunch of single cell data and bulk RNA sequencing data. And, you know, as many people in the audience know, GD2 is the proven immunotherapeutic target in neuroblastoma with an FDA-approved immunotherapy. And you can see that there may be problems with these therapies because there is expression in central nervous system and some other tissues. And there are differentially expressed genes, but what we would really like to go after are genes that are highly expressed in all neuroblastomas and very low in normal tissues. And again, I'll come back to these FOX2B, FOX2A, those things I showed on the prior slide. Those transcription factors are there, but there are also some cell surface proteins that are in this sort of differential expression range. And so I'm going to tell you one story about a cell surface receptor and then another about this FOX2B. So I just very briefly want to talk about GPC2. This has been a great collaboration with Crystal and Robbie and Sabine, and largely Sabine driving the work on the Stanford side. This was a paper that was one of the first that emerged out of our initial Stand Up to Cancer St. Baldricks efforts. And this was by Chris Bossi, who is pictured giving a lecture about this at CEOP back when we could travel, and back in 2017. And so instead of going through all the data, because it is published, it's been published for several years now, I just want to discuss the main important points. And these were all things that we considered essential if we wanted to develop an immunotherapy that should be hopefully effective, but also not toxic. That the expression in tumor tissue appears to be very high and appears to be homogeneous. And Crystal and I have had a lot of debates about this. But compared to other cell surface targets, we think that it has high homogeneous expression. It really has restricted normal tissue expression, and I'll show you data on that. This is, I think, one of the most important points is that GPC2, not much was known about until Chris came across it. And it is a MYC-driven protein, so it is a MYC gene. And it's also on a chromosomal locus on 7q that is very commonly gained, and expression is related to copy number. And Chris showed in this paper that if you deplete it genetically, cells don't tolerate that and die. So we think that this is a non-mutated oncoprotein, a theme that we've been developing. He defined the mechanism of overexpression and discovered a binder, an scFv, that is very specific for this protein. And so these are some unpublished data, a paper that was just submitted recently, that here is the crystal structure of the GPC2 protein. And then this is this so-called D3 binder. This is the antibody fragment. And you know, you can see if you can rotate, I didn't have the rotating thing here, but it's binding a conformational epitope. So if you look at the map of the GPC2 protein, just lining up the amino acids, it binds here more in the N-terminal region, and then here more in the C-terminal region. And what Chris has showed, and I'm not showing the primary data, is that if you do have normal tissue expression, and there is some in the testes, and perhaps a hint of it in the esophagus, you express a isoform that only includes exons 5 through 10, whereas tumor always expresses an isoform that includes all 10 of the canonical exons. So we think that gives some tumor specificity. When you take this antibody and put it on a membrane protein array, so this is over 6,000 proteins, to look for off-target effects, GPC2 is the only signal. It's very clean. And then what Chris had done in the original paper and extended recently is developed an antibody drug conjugate against this that shows this sort of potent efficacy in both neuroblastomas as well as small cell lung cancers, which overexpress GPC2 as well. And you can see the different doses there. You know, a single 1 milligram dose, you get a response, but it's not sustained. But as you give a little bit more, you can get sustained complete responses. So for reasons I'll come back to in a second, we think a CAR is actually probably a better tool for this protein than ADC. And Sabine really has done beautiful work and did not get discouraged when the early work was not as positive as their final story, to develop a highly specific and effective CAR T therapy. So I'm not going to get into the details of this, and Sabine just submitted the paper and it's out for a review right now. So we have our fingers crossed. But to make a bottom line, the bottom line is that substitution of the 28 transmembrane domain was really what was essential to get curative CAR T efficacy in these mouse models. And, you know, as expected, the 4-1BBs persist a little bit more, but it's the 28 transmembrane domain that is really, really important. And that may be due to lower antigen density. It may be due to maybe heterogeneous antigen density. And that's something that we're still working on, but it's very clear that that engineering was essential to get a highly effective CAR. And the reason why I think CARs are going to be the good format to go forward is because while we concentrate on neuroblastoma, this is RNA sequencing data here, 4,000 different neuroblastomas that shows that GPC2 expression is high in most. So this is showing that percent of tumors with high GPC2 expression. So it's not all, but it's many. And that the red is the median expression level in RNA sequencing. So this is high, it's 70 or so. But there are many other neural-derived cancers that have high GPC2 expression. So, and since many of these are behind the blood-brain barrier, we think that CARs, if we want to have a broader impact than just neuroblastoma, CARs are likely the way to go. And so, you know, thanks to Sabine's work, we're moving that, and Chris's work, they're both working on slightly different CAR structures. That's happening. So For GPC2, I think it's very clear that it's an important oncogene in this disease. It has highly restricted expression that I didn't show you an example of today, but it's in the original paper. And that these, the CARs that Sabine and Crystal engineered that are tuned to low antigen density, as Robbie showed in a recent beautiful paper, show potent efficacy in mice. And so we do have a phase 1 clinical trial of this planned and are in the late stage of the IND enabling studies at CHOP. And the study would start at CHOP for the dose finding safety phase, and then hopefully we'll expand into the pediatric CITN that Crystal runs in the not too distant future. So other than finding the targets, neuroblastoma is famous for being probably the coldest cancer known to man. So we published, and many other people did, that mutation burden is low, that Rene Bernard's had this paper back in 1986 where he showed that when you have MYC overexpression, you have low class 1, and that's something that now has been proven more elegantly by this Australian group showing that neuroblastoma and small cell lung cancer are lowest for HLA expression and a lot of the antigen processing pathway, and that we and others have shown that if you just chop up a neuroblastoma and look for T cells, you don't find many or any. And so that didn't stop Mark Yamakovich from postulating that we could get after targets that are considered undruggable by going after this with this sort of pipeline. So what Mark did while he was a graduate student in the lab, he's now graduated and is, thankfully for me, doing a postdoc with me, is he performed the things that my lab can do very easily, which is next generation sequencing, DNA sequencing, RNA sequencing, with a very small number of tumors, 8 patient-derived xenograph tumors, and then 8 primary neuroblastomas, all from high-risk patients. And in parallel, he did MHC pull-down and mass spectrometry, which we dubbed and others have dubbed immunopeptidomics. And Mark leveraged, he did this in collaboration with a group in Tübingen, Germany, and leveraged their sort of, yeoman's-like effort of going painstakingly through a bunch of normal organs and doing the same peptidomics experiment. And I'll come back to that and how that's been so useful to us. And then this has led to us finding a lot of peptides that we think are interesting. We have a great collaborator with my next door neighbor, who is Nick Skarakis, who is a very talented biochemist and expert in peptide MHC structural biology. So he can quickly crystallize peptide MHC complexes that we're interested in. And then, as I'll show you, we can engineer different therapies. I'm going to concentrate on CARs, but we can also do TCRs depending upon what the peptide is and how we're thinking about doing cross-reactivity screening prior to getting into the clinic, so we don't run into toxicities in these kids that we could have predicted. So this is summarizing what the data looks like. So Mark went to Germany twice. First, he packed along 8 PDXs in a suitcase, and he did the pull down there and eluted about 500,000 peptides. And the first thing, which was predictable but was pretty stunning, and there was 0 neoantigens, right? So these 8 PDXs we sent, it's not that they didn't have any mutations. So there were 3 or 4 that had an ALK mutation. There were a couple with some RAS mutations. But, as we predicted, these, very seldomly get out to the cell surface in an MHC groove for a lot of different reasons. And so, but when you take these peptides And then you filter them down quite quickly, just, which ones are predicted to bind with high affinity, which of these are differentially expressed, just looking at bulk RNA sequencing data. And then finally, the last check of which are these are absence, they had to be absent in the entire 190 tissue normal ligandome. You get down to a small number of peptides that we think are, highly specific to neuroblastoma. We prioritize 4 because these 4 are all involved in that sort of epigenetic core regulatory circuit. And I've alluded to FOX2B a couple of times, and I'll come back to this because this is where we focused. But these are this after the first experiment, we got, pretty excited about this. And Mark packed up 8 primary neuroblastomas and flew back to Germany and did this experiment. So this is just a different way to look at the data. And so this is, there's just 8 tumors, and this is the percentage of tumors that showed a particular peptide, and this is the normal ligandome data. And so because primary tumor biopsies in neuroblastoma are very small, he had many less peptides because the number of peptides you get scales linear with the mass of the tumor that goes in. But he alluded, this number of peptides. And here, 100% of the neuroblastomas had a peptide here, but with about 20% of the normal tissues had the peptide. So obviously, we're not going to look at any of the peptides here, and really concentrated down here the peptides that were exclusive in neuroblastoma. And so there were about 4,000 peptides from 2,800 genes that were not observed in the normal tissues. And if you do a gene set enrichment analysis of these antigens, it only returns one term, and that term is highly statistically significant, and that is sympathetic nervous system development. So that really tells you that these antigens are lineage specific. I don't know how my thing got on the automatic go. And really concentrate on these that are recurrent, and many of the ones we'd seen previously in the PDXs we saw again, and prioritize these 6 peptides for further development for potential immunotherapies. So I've mentioned FOX2B a couple of times. I've thought about this protein for a long time. Jean-Emile and Olivier Delattre scooped us back in 2004, where they wrote this beautiful paper showing that there are occasional patients with neuroblastoma who have germline mutations in this FOX2B gene that cause that was associated with neuroblastoma. I happened to be caring for this little kiddo right here at this time who had this very significant family history of neuroblastoma and Hirschsprung disease. And we were able to show pretty quickly that this family was also affected by a germline FOX2B mutation. We've done a lot of biology to show how important this gene is. And this is, the expression in 189 neuroblastomas. And then this is the GTEx data showing this really clean expression in thousands of normal tissues. And then many of you are probably aware of this really nice data set looking at gene expression over time. And so there is FOX2B expression, for example, pretty high FOX2B expression in the cerebellum and in the developing fetus, but by the time of birth, it's off. And so every tissue, it's off, and as shown in these data. And this is just some ChIP-seq data from my lab that's fuzzy for some reason, I don't know, that just shows that there's a super enhancer and that all of these transcription factors are binding in the promoter region, and is sort of a feed forward loop. So there's a lot of underlying biology. So how do you get binders to peptide in MHC? Well, we spent a lot of time with our longtime collaborator, Dmitry Dimitrov, doing this, depletion strategy. So he has a beautiful, fully human scFv library of 10, it's a 10 to the 10th complexity. And we, depleted this library first with a single peptide, irrelevant peptide in the MHC to get rid of things that were just binding to the to HLA. This I should have mentioned. This FOX2B was on HLA-A24, so A2402. So that's the MHC peptide refolding that we did. And then after this depletion, we did enrichment. And, we got a lot of binders like this that, would bind to FOX2B on the MHC, but would also bind to an irrelevant peptide, a decoy peptide. And we got excited about this sort of, this looked pretty specific. I'm not going to show any of the data because it will be 2 years of reminding me of the misery. But this little bit of binding to the decoy was enough to get, cross-reactivity to the MHC. And Mark did an incredible job of mutinizing these and getting them more specific. But we never could get them specific enough to, in our opinion, to take them to the clinic. This was rescued really by an amazing collaboration we have with a company called Affinity Bio. They're outfitted in near Melbourne, and they have a really interesting bacterial base and flow sorting mechanism to do panning, and the fact that it's flow based allows them to not only do the screening, but to do a lot of counter screening to make sure that you have specific. So this is, one of this 10LH I'll be coming back to a lot. It has this sort of binding characteristics. The green is a FOX2B and red is an irrelevant peptide, a relatively slow on rate, but then a very slow off rate. You can see here that the binding to FOX2B is very low nanomolar affinity, and all of these other 895 other what we call counters or other peptides, there's no binding, and you can see that it has a really good footprint. So this is an alanine scan, and you can see the amino acids for the 9-mer, except the 2 anchor residues, and this is what we consider a very good footprint, reducing the binding with a single mutation very significantly. And then we can map these onto the crystal structure to understand what residues, so this is looking down on the MHC, and then over on the side, what residues are really critical for the interaction. And as I'll end, this comes to the last experiment I'll show you is, are we really like a TCR restricted to HLA A2402, or could this peptide in the binding to the peptide be specific enough that this could be recognized on other HLA alleles? And we think the answer is yes. And I'll show you a little bit of data. All of that's ongoing. So this is, the kind of cars we were getting originally, where there's, this is the FOX2B and then a cross-reactive HLA-A24 peptide. We were getting this sort of cross-reactivity. We spent a lot of time mutagenizing these to try to get them to fall down here. But this is that 10LH binder, another binder we prioritized from Affinity that showed really clean and specific binding to the FOX2B HLA-A24 dextramer. Mark and Alvin created this really nice algorithm to predict cross-reactivity. And it's a cross-reactivity antigen prediction. They want to call it CRAP. And I'm trying to discourage them to come up from that and come up with another acronym. But this is how the algorithm looks. Here's FOX2B, and then these parent genes have these peptides that are predicted to bind HLA-24 with a high enough affinity that they might be recognized. And then this column shows whether or not it's been found in the normal ligandome or not. So theoretically, something like this might be of concern. Went ahead and made all these peptides and expressed them and expressed the 10LH CAR in jerkhead cells, and then did this flow. And you can see the FOX2B is very specifically bound. Most of these are very low relative binding, but there are a couple that are kind of concerning. There are these two right here. However, this is in a FOX2B negative colorectal cell line that is A24 expressing. And we're pulsing these cells with peptide. So with no peptide, you don't get any killing. With 0.1 micromolar of peptide, you get very specific killing. And then for these two peptides, pulsing with these peptides, you can see no killing at 10 micromolar, and then there is killing at 50 micromolar, which we think is super physiologic and not relevant, whereas for this one, there's no killing at all at either 10 or 50 micromolar. So we think that this is a safe CAR to take forward. In terms of efficacy, we've done, I think I need to get rid of my pointer for a second, I'm sorry. The two cells on the left are neuroblastoma cell lines. The two cells on the right are melanoma and colon cancer. And this is a caspase assay, time lapse over 2 to 3 days. And you can see that the neuroblastoma cells all die at an effector at a target ratio of 1 to 1. Whereas there's no killing in the control cell lines. This seems to work very well in vivo as well. This is a cell line derived xenograph, HLA-24. And then this is a HLA-24 patient derived xenograph. This one was not properly controlled because we didn't have enough T cells, another pandemic blue sort of situation, but 2 different binders and then just a sterile PBS, which, that's the normal growth kinetics, and you can see complete and sustained responses before the mice develop GVHD. This line is a new line in our lab, and it is probably the fastest growing PDX that I've seen. And what was remarkable here is that some of the mice who got the CARs exceeded our 2 centimeter IACUC size volume and needed to be sacrificed, but that was only about a week after we gave the CARs, and then they all came down. So this line was so fast growing, it's sort of outran the T cells for a while until they expanded enough. So we have other ongoing experiments that are still earlier on that are showing equal efficacy. So we're quite happy with this. The last thing I wanted to mention is that, we do think that we may not be as restricted as a TCR if you have a highly specific SCFV to the peptide in the groove. Mark and I have published this algorithm that we used both for a cancer purpose and also a SARS-CoV-2 purpose to look at population scale prediction of different peptides. So this is showing an output from the algorithm of the FOX2B protein from N terminus to C terminus here, and all the different peptides, and then the frequency with which that peptide should be presented at high affinity across the population. So what this is saying is that this peptide, which happens to be the FOX2B peptide that we're interested in, is on A24, that's where we discovered it, but is also presented with high affinity on these other HLA alleles. And so with Nick, we can model these, and we actually are creating the crystal structures of these because we really want to understand the biochemistry of this. But the question is it presented in similar enough a fashion to be recognized? And the answer is maybe. Or potentially yes. So here's A23, and you can see CD19 no binding, and then here are 2 CARs, and the 10LH for whatever reason binds stronger than the 302 CAR. And then this is another HLA allele, and you can see the same thing. Sorry, the font is, I didn't fix up the font here, but the same thing where we're seeing binding, and we're also getting killing. So this is NBSD and SKNFI. They're both A23 lines. And I'm not showing the time lapse here, but this is after 48 hours, E to T of 1 to 1, we're getting complete killing. And then in this NBSD cell line xenograph, that is HLA A23, you can see that the CAR that binds better is causing complete and sustained regression. So we think that technology now allows us to go after what we've largely considered undruggable key transcription factors in this disease, neuroblastoma. And we're very interested within our network to see if we can generalize this concept to other human cancers. And with the group at Stanford, we're already planning experiments in Ewing sarcoma that will be done at Stanford, and hopefully Mark and I can help out with. And we have gotten funding through CHOP to bring this to a clinical trial. So the IND enabling studies are ongoing, and we'd love to be able to get proof of concept that what we're seeing in the mice can actually help a child. So there's a lot of people I'd like to thank on the CHOP and Penn campus. It's just been my pleasure and privilege to work with Crystal over the years, both in the St. Baldricks project, where we have over 100 investigators, and very importantly, and it was a decision, that we made early on, is to really embed advocates throughout the program and really have them teach us important things that we need to be thinking about as we develop these therapies for children with really high-risk diseases. And in this, in the Immunotherapy Discovery and Development Network, this is the Bo Biden and other collaborators. And then, the classic Zoom lab meeting shot that I'm really hoping that we can do away with these and have actual pictures in gardens and other things like that very soon. Really want to show Dave, who's done the mouse work. Quinlan's done a lot of the cross-reactivity work. Mark, who I mentioned, has been leading this work. And Nathan and Alvin, who do a lot of the computational work. So thank you for your attention.
Thank you, John. That was fantastic. So if you want to ask a question, please put it into the Q&A feature. And it looks like we have some time. Maybe I can start with a couple. So you told us the sort of classic, showed us the classic paper showing, low MHC or absent MHC on neuroblastoma that we all hear about. But now we're targeting, or you're targeting an MHC restricted peptide. So what is your thinking about whether MHC expression will be rate limiting and how one might be able to modulate that?
Yeah, it is the potential Achilles heel of the whole peptide MHC, is that you have really led the way in teaching the entire community of the importance of antigen density in order to trigger cell killing and expansion. It's critically important. I should have shown this SKNAS, which was the first one we did. We handpicked because for whatever reason it happens to have the highest MHC expression, but SD, and that we picked that COG line because it's actually, if you just look at the RNA-seq data and flow, it's actually quite low in MHC, more reflective of, I think, the average patient in the clinic. So we're seeing activity. It's a mouse. It's a model. It's wrong, right? So we'll have to see when we get to the clinic. We are, it is very clear that the mechanism of MHC downregulation is poorly understood in this disease, but that the PRC2 complex does play a very important role in the downregulation. And we have data that in NMIC amplified neuroblastomas, you can dramatically upregulate MHC in a dish, at least, with EZH2 inhibitors. Interestingly, we still don't understand this. Many of the neuroblastomas that are not harboring NMIC amplification have overexpression of c-MYC. And in those models, we actually see the MHC go down with EZH2 inhibition. And so it's not going to be a simple answer, but I do think that we have to really, my laboratory and our entire network really need to think about ways to modulate MHC in our patients that are tolerable and will not counteract the T cells we're putting in, which is always the worry when you start messing with small molecules and that sort of thing.
Yeah. I guess another question would be, have you also looked at TCRs and any thoughts about relative potency or the challenges, and maybe it's easier to make a CAR than a TCR, but the choice between targeting an MHC peptide with a TCR versus a CAR?
Well, we think that for these non-mutated self-peptides, where there's going to be some tolerance, that they're not going to be as immunogenic. And so we think that for these self-peptides, going after them with CARs, and now that we have the technology, I think to really get something that's just getting the peptide, which is, thanks to our friends at Affinity Bio. But for a neoantigen, whether it's a point mutation or it's a splicing defect, I think that TCRs are going to be the way to go there. And we have not been able to get to much of that yet, but I think that in your Ewing project, for example, I have a feeling that you'll probably come up with a lot of non-canonical peptides where it's going to probably be better to go after it with a TCR.
Interesting. Okay, so question, how does antigen heterogeneity affect MHC presenting peptides?
It's a good question that I don't have the answer to. We could probably approach that sooner rather than later in terms of regional heterogeneity. But I don't know that we're quite at the point where we could do this in a single cell experiment or something like that from a single biopsy. But we know that heterogeneity is the Achilles heel, and it's something we're going to have to pay a lot of attention to.
Raj Rahati asks, what is the mechanism by which GPC2 overexpression drives oncogenesis?
So what we know is that the proteins that are overexpressed directly from NMIC often have important roles in proliferation, immune evasion, or anything that makes a cancer cell. And so to me, the fact that it's a MICN target gene and it's on a chromosomal locus where there is, the cancer goes, so when you don't have NMIC amplification, you very commonly have gain of the GPC2 locus and overexpression. So the cancer has gone through some machinations in order to upregulate GPC2 and keep it up. Exactly how it's orchestrating that is actually the focus of Chris Bossi's K08 award. And it's clear that that GPC2 orchestrates signaling through the Wnt beta-catenin pathway. And we think that's interesting because that pathway is never mutated in neuroblastoma, whereas it's mutated very commonly in many other childhood cancers. And so we think that's the major mechanism, but I think that maybe Crystal can invite Chris out in a year or two and he can dive really deep into that.
Okay, that's all the questions I see. Anybody else have any? Otherwise, I think we'll give people 10 minutes back. And thanks, John, for his time. It's been a pleasure. Thank you, everyone, for attending.
Thank you, everyone. Bye-bye.
Take care.